if you'd like to record this. Okay. This is kind of an unusual class. When I was your age going to school, we didn't start with a half-hour meditation, so that's kind of cool. That's a good thing. We get us in the mood. I hope that nobody is so relaxed that they fall asleep, which is tempting at this time of the afternoon. But hopefully the lecture will be sufficiently stimulating. So we'll get started, because there is a lot of material I'm hoping to cover, and we'll see how far it goes. So if my screen share is enabled, yeah, you've got to let me do screen share, and I'll open up this lecture here. And where are we? Wait a minute.
Should be set up.
No, I got it. Okay, so you can all see this, I hope. And so we'll get started. Just a second, let me disable or make the tools invisible here, so that does not hide floating meeting controls. There we go. All right, so we have a full picture here, and the name of this lecture, as you can tell, is a pharmacognostic survey of mind-altering natural substances. Does everybody know what pharmacognosy is? Anybody who knows what it is can use your virtual tab to raise your hand. Not that it really matters, but pharmacognosy is the scientific interdisciplinary investigation of natural products, biologically active natural product. Pharmacognosy used to be taught in many pharmacy schools, but it's no longer taught widely, because back in about the beginning of the 80s, they assumed that natural products, that nature was not going to be a significant source of new medicines. So the pharmacy programs dropped pharmacognosy, which is a shame, and they were totally wrong. There are many new natural products that have come out since that are important medicines, and including psychoactive medicines. So want to give you before we get into the sort of materia medica of this lecture, I want to give you a very quick, brief sort of detour into neuroscience, pharmacology, and chemistry. So this is a depiction of the human genome, one way of depicting it, and it shows the different sections of the genome and the functions that they're devoted to. Interestingly, you can see almost a third of it, a little more than a third of the genome, molecular functions are unknown. So these genes are not 41.7% of the genome is unknown. But the part that we're interested in is the section that's about signal transduction. And signal transduction is a process of communications within the organism that is molecularly mediated. It involves molecular messengers that are binding to different types of receptors. And essentially, signal transduction is the process that organizes a living thing in space and time. It, if you will, it choreographs the dance through time and space that makes an organism, which is not really a thing, it's a process that orchestrates it through time and space. And that's all elegantly regulated by signal transduction processes. So signal transducing molecules represent about 1/8 of the genome, and the receptors for those molecules represent about 1/20 of the genome. And of the various kinds of receptors that are important in this process, particularly in psychopharmacology, the so-called G protein coupled receptors, the GPCR, represent about 2% of the genome. These receptors are by and large what psychoactive drugs interact with. And then the other one is the transporters, about 1.7% of the genome. Things like the serotonin transporters, the dopamine transporters, the essentially the membrane pumps that reuptake the neurotransmitters from the synapse. And those two are very important targets for drugs, which we'll talk about. So the brain's signal transduction functions are mediated by these neurotransmitters, and by and large, neurotransmitters are small molecules that mediate this crosstalk between neurons. Really, the brain is a machine for signal transduction. And all of its critical biochemical functions, including our experience of conscious awareness, are mediated by this neuronal communications network. So just a quick look at neurotransmitter phylogeny. I wasn't sure whether to put this in here, but it might be useful to orient you. So there are various classes of neurotransmitters, and the ones that we'll mostly be talking about are the small molecules, things like acetylcholine, the so-called monoamine neurotransmitters like norepinephrine, epinephrine, dopamine, serotonin, and so on. We're going to be talking today a lot about serotonin and to some degree acetylcholine. And then there are the others, the amino acid small molecules like glutamate, GABA, glycine, and the purines, adenosine, and in its derivative. Adenosine, for example, is very important in the pharmacology of coffee, which we're actually not going to talk about because it's too pedestrian and well known. We could say a lot about it, but we're not going to discuss it. The other class, another class, are the peptides, which are generally small peptides like the opiate, the endogenous opiate peptides, enkephalins, dynorphins, endorphins, and so on. We're not going to talk about much of those except one thing a bit later. And then there are ions. There are actually neurotransmitters that are gases or ions. These are important. And then other, and among the other are the cannabinoid receptors, the receptors for things like your endogenous, your anandamide is an example of a cannabinoid neurotransmitter. So just kind of to orient you, there are many, many kinds of neurotransmitters, and they don't occur only in the brain, they're all over the body. So, it's a very complex picture when you start talking about neurotransmitters and the various ligands or substances that bind to them, some of which are endogenous and some of which are not. So that's the neuroscience. That's as far as we're going to go with that. But then let's talk about plants and plant chemistry. Plants have mastered this kind of miraculous process of photosynthesis. They have figured out how to harvest sunlight, use the energy of sun through different molecules that are able to absorb that solar energy and use that in the manufacture of great variety of organic molecules that they make from the precursors of sunlight, water, and carbon dioxide. And in the process of doing this, they exhibit some very important regulatory functions in the ecosystem. For example, plants are what is mainly sequestering carbon dioxide out of the atmosphere. They can absorb carbon dioxide, use it in the manufacture of complex organic compounds, and remove it from the atmosphere. Of course, the problem that we're facing now with global warming is the fact that we're emitting much too much carbon dioxide and the plants can't keep up. It doesn't hurt either that we're burning down the rainforest. So rainforests, especially the Amazon rainforest, for example, is an enormous carbon sink. But not only are we cutting it down and not getting that function, but we're also re-releasing all the carbon that those plants have sequestered over millions of years. So, it's a very stupid thing to do. We're very stupid animals in some respects, and this is having an enormous impact on the environment. Another cool thing about photosynthesis, many cool things about it. But another important thing is one of the byproducts of photosynthesis is oxygen, and oxygen happens to be what we breathe. So, it's a neat balance, and we can thank the plants for producing the atmospheric oxygen and maintaining the oxygen at those levels, which is about 21% of the atmospheric gaseous composition. So plants have figured this out. They use solar energy, carbon dioxide, and water to make these simple organic compounds, starting with simple sugars. And then from there, I think you could all see my cursor. Then from there, it spins off into different pathways. And some of these are pretty much universal in life. They're found in all living things. Those in the blue pathways are pretty much the molecules of life. But those in the yellow boxes are so-called secondary products. These are plant chemicals that plants make, and they're not essential for life, because if they were, they'd be in all organisms, but they're useful. They're only found usually in certain families, certain genera. Their distribution is wide in the plant kingdom, but not universal. And so what do they do with these molecules? They have various different classes. One of them that's particularly important are the nitrogen-containing secondary compounds. That includes things like alkaloids. Alkaloids are very important sources of natural medicines. Alkaloids tend to be biologically active. They're either toxic or psychoactive, or they have different types of important biological activity as poisons, for example. So many of the drugs, all the bad drugs, no such thing as a bad drug, but all the drugs we think of as bad are probably alkaloids: nicotine, cocaine, heroin, those sorts of, well, not heroin. Heroin is semi-synthetic, but morphine that it's derived from it is an alkaloid. Another important class of secondary compounds are the terpenes. And they vary in terms of their molecular weight. The lower molecular weight terpenes, the so-called monoterpenes, have about 10 carbons. And these are the plant aromatic constituents. This is where plant fragrances come from. And, they are also biologically active. So there's this, each one of these boxes in this diagram represents thousands upon thousands of different kind of molecular structures. So it's a very simplified diagram. Well, if these secondary compounds are not essential for life, what do the plants do with them? Why do they make these compounds? Well, the secondary compounds are messenger molecules. A famous botanist once said, plants substitute biosynthesis for behavior. Plants are stuck in one place. Animals react to their environment through behavior, the so-called fight or flight reaction. They can run away from danger. They can respond to danger by behavioral adaptations. Plants are stuck. They just sit there. So they have to figure out better strategies for optimizing their relationship with other organisms in the environment. And they use chemistry to do this. If plants have a language, that language is chemistry. And they use these messenger molecules to mediate their relations with other organisms in the environment, other plants in their neighborhood, fungi, microorganisms that might be associated with them or might be threatening them, insects, of course. Insect pollination biology is entirely mediated by plant secondary compounds. Herbivores, things that might want to nibble on plants, sometimes they produce compounds that are repellents, that don't taste good or that are poisonous if they want herbivores to stay away. Other times they produce compounds that maybe smell good and taste good, because in some instances, it's to the plant's advantage to be consumed, like in seed dispersal and that sort of thing. Well, humans are among these organisms that might want to nibble on plants for one reason or another, and sometimes they nibble them because of the effects they can have. So these secondary compounds basically have a spectrum of effect. It's either defense, which like in the case of repellents, where the message is stay away, I'm a badass, you don't want to mess with me, just leave me alone. That can be one of the messages that these chemical messengers put out into the ecosystem. More often and more interesting, it's a more complex message. It can be semiosis, often coupled with symbiosis. Semiosis is sending a signal. And in other words, a signal which could be a fragrance or a color or some sort of sensorily detectable stimulus that an organism, an insect, a person, an herbivore, a fungus, whatever, can sense and either get the message, stay away, or come closer. Come closer. Let's do stuff together. Let's do something together. And then you get into the realm of symbiosis, which comes collaboration with plants. Come hither, let's form symbioses. And if, it used to be that evolutionary biology was thought to be entirely about competition, survival of the fittest, and that sort of thing. But that's sort of an obsolete understanding now. A symbiosis is found at all levels of nature, and forest ecosystems, for example, could not maintain their health unless they had the symbiotic relationships with the fungi in the soil that form these mycorrhizal relationships with the roots, and these old growth, enormous old growth trees and old growth ecosystems are just networks of these symbiotic relationships that act on the ecosystem level very much like a nervous system. They don't look like a nervous system, but like a nervous system, like brains, they're based on signal transduction processes. There's a wonderful book that has been out for a while about this called Finding the Mother Tree. I highly recommend it. I'm not going to say any more than that, but it's definitely worth a read. It's by a UBC forest ecologist, and it's really kind of revolutionized the field. So when it comes to the interaction of these secondary compounds with us, with our brains and our nervous system, the surrealist painter, someone asked him once, how do you get these ideas for these crazy wild paintings that you produce? Are you on drugs? He said, no, I do not take drugs. I am drugs. And there's great wisdom in that. We are basically biochemical engines that run on drugs. We run on these neurotransmitters, signal transduction molecules. And the brain neurotransmitters and plant messenger molecules all came from the same evolutionarily precursors and probably served similar functions. So the same compounds that may be neurotransmitters in the brain may have originally evolved to propagate signal transduction on the ecosystem levels. So it's not surprising, given this evolutionary commonality of origin, it's not surprising that plants contain a panoply of neurotransmitter-like compounds that act on brain receptors. In some cases, the plant compounds are identical to neurotransmitters. More often, they're analogs. They're similar to neurotransmitters, but not exactly the same. So this is the situation. And most psychoactive drugs, it says, function by affecting the synthesis, storage, release, degradation, or reuptake of neurotransmitters, or by blocking or mimicking their effects at synaptic receptors. So that's the neuroscience, a little look at phytochemistry and plant interactions. So now we'll go into looking at some of the different classes of these compounds. I've divided it into a couple of different sections called shamanic medicines, because most of these compounds and plants that we'll be discussing find a role. They're used in traditional societies, indigenous societies, in shamanism, which is kind of a combination of ritual and medicine. Everything comes together. It's probably the most ancient practice that we have. It's really what medicine as well as religion originated from. And we can talk about in under this rubric of shamanic medicines, we can discuss classical psychedelics. And that's what this first section is about. I think it's a convenient category. I like to talk about true psychedelics. You could talk about true psychedelics that are distinct from other classes of psychoactive drugs. The true psychedelics interact with the neurotransmitter serotonin. Other psychoactive drugs may resemble psychedelics, may have profoundly may be able to induce profound altered states of consciousness, but they're not true psychedelics in this kind of restricted pharmacological sense that I'm using the term. For example, the empathogens, things like MDMA, is psychoactive for sure. It has therapeutic potential, but it is not, strictly speaking, a true psychedelic in this sense. Another example are the tropane alkaloids from Datura and the other nightshades. We'll be discussing those downstream. Again, profoundly psychoactive, not psychedelic. So what distinguishes a true psychedelic is its interaction with serotonin. Serotonin is one of these neurotransmitters. It is a tryptamine, so it's called 5-HT, 5-hydroxytryptamine, and it's one of the master neurotransmitters in the brain. It regulates processes such as sleep, dreams, attention, perception, mood, sexual behavior, eating behavior, and of course, perception. Other important neurotransmitters that you're all probably familiar with, at least in concept, are also monoamines like dopamine and norepinephrine. And the serotonin networks originate in the brainstem and innervate the major brain areas known to be linked to conscious experience, such as the limbic system, the hypothalamus, and the neocortex. And there are about 14 different subtypes of serotonin receptors in the brain, and the 5-HT2A receptors are the key receptor in the action of psychedelics. So this diagram here, we've totally understand, or not we, but scientists have worked out very complete understanding of these 5-HT2A receptors, other neurotransmitter receptors. They know the secondary and tertiary structures. They know what they look like. And they even know to a certain degree how some of these molecules bind into the receptor itself and elicit an effect, whether it's serotonin or a drug. This particular diagram shows LSD binding into the receptor pocket of the 5-HT2A receptor. So that's, most of the true psychedelics, in fact, by definition, the true psychedelics do work on these 5-HT2A receptors. Now, it's important to realize that as a rule, drugs are not selective for just one receptor. They usually affect a variety of receptors. For example, LSD is a good example of a drug. It works primarily on the 5-HT2A receptor in terms of its psychedelic effect, but it also affects other serotonin receptors, dopamine receptors, and so on. So LSD is a good example of what they call a dirty drug. It's not a entirely specific receptor-specific compound. Most drugs are dirty drugs to a certain degree. Very rare exceptions. We'll talk about one a little bit, but as a rule, that's not the situation. So if we look at these chemical classes of the classical psychedelics, the true psychedelics, they belong to one of three large classes: the phenethylamines, of which mescaline is an example, the lysergamides, of which lysergic acid amide, or LSD, are examples. LSD is not a natural compound. This is a natural compound, very closely related to LSD in its structure. The tryptamines, like dimethyltryptamine, and then the beta carbolines are a fourth class. These are not in themselves psychedelics, but they're monoamine oxidase inhibitors. So they can orally activate DMT and other tryptamines, which are normally not orally active. DMT, for example, has to be injected or smoked or otherwise injected other than orally, because if they're eaten, the enzymes, monoamine oxidase enzymes in the gut will just inactivate them. But if you take it in combination with a beta carboline, then it's protected and it becomes orally active. This is the basis of ayahuasca pharmacology, which we'll talk about a bit downstream. So starting with the phenethylamine derivatives, of which mescaline is the chief example. These are found in the Cactaceae, the cactus family. And as far as anyone knows, they don't occur outside that family. And they're the main alkaloid, the main psychedelic alkaloid in peyote, and also the Trichocereus species in the Andes, which are now called Echinopsis. So Trichocereus or Echinopsis are at the basis of very ancient traditions of shamanism in the Andes, 8 to 10,000 years ago. And peyote, similarly, quite ancient. There are archaeological sites dating back to 8,000 years for peyote in the southern Texas region. So these have been around for a long time, and they are, basically among the oldest shamanic medicines in the New World. And mescaline is found in other species of cacti, actually about 30 species or so of cacti, not just the Trichocereus and Lophophora, but these species here also contain mescaline. Interestingly, many of these cacti are beautiful plants, so many of these plants are sold as ornamentals in the nursery, in many nurseries. And well, you didn't hear it here, but you could just go there and purchase these and grow them in your garden if you like, or do other things that technically probably would be discouraged, shall we say. But there it is. And mescaline is a very interesting compound. Primarily, one of the reasons it's interesting is because it's so simple. It's a very simple molecularly speaking, it's a very simple compound. So it's a good candidate for medicinal chemists to play with. And that's what medicinal chemists like to do. They like to take a simple, relatively simple molecular scaffold and tweak it and make structural variations and then see how that changes the activity. This is basically what medicinal chemistry is all about. Mescaline resembles ephedrine, which is one of the natural stimulants. For example, in ephedra, the mahuang, the Chinese medicine, or in khat, the leaf from Catha edulis that people choose. So it has a amphetamine-like effect. Essentially, it's a natural amphetamine. MDMA is an example of a semi-synthetic analog of mescaline, and it has the properties that it has. It's structured. It works on serotonin, but it's not a true psychedelic. It doesn't work the same way. And then there's potential to make many, many analogs of mescaline, and this can be done. For example, you can take the methoxy group here and put it up here. So you've got a 2,5-dimethoxy configuration. And then X can be just about anything. If you make it a methyl, you have a compound called DOM, which back in this day on the streets, this used to be called STP. It's about 100 times more potent than mescaline. It lasts, depending on the dose, anywhere from 12 to 36 hours. And then you can actually halogenate the X. You can use iodinated radioactive iodine or radioactive bromine compounds, or they don't have to be radioactive, but the radioisotope labeled compounds are very good for brain imaging studies and that sort of thing. And so they're medicinally useful. DOI is maybe one of the most 5-HT2A selective molecules that is known. And so, even though these are psychotropic, they also have their uses in neuroscience and brain imaging, even, that have little to do with the fact that they are. It's all about how they're uptaken into the brain and distributed through the brain. We have different techniques for mapping the distribution of serotonin 2A receptors. That's what I was doing when I was a youngster working on a postdoc at NIH. I had radioactive forms of DOI that we could use to map these networks of serotonin 2A receptors. And one of the reasons I was able to do this is because I had a relationship with Alexander Shulgin, who's a medicinal chemist who is a pioneer in this whole area of looking at the structure-activity relationships of the phenethylamines, is what he concentrated with. What he concentrated on initially. He, over the course of his career, created about 300 different kinds of mescaline analogs. And if you're familiar with his writing, he wrote 2 books, one of which is called PCOL, Phenethylamines I Have Known and Loved, and the other one is called TCOL, Tryptamines I Have Known and Loved. They're both well worth reading. They're very interesting, and both of them are written. The first half of the books are kind of a lightly fictionalized story of his life, but the second half of each book are the recipes for the compounds that he made, and his trip reports, because he evaluated personally every one of these. Along with some friends that were obviously experienced trippers, and kept careful notes. And it's really been it's been an important contribution to neuroscience. In my opinion, he should have been given the Nobel Prize, but because the work he did was so controversial, that was never a possibility. And then another pioneer in this field is Dr. David Nichols, who is the founder of the Hefter Research Institute. And he continued a lot of the work that Shulgin did. He was a professor of pharmacology and chemistry at Purdue University for many years, one of the few investigators who actually got government funding for his work on psychedelics back in a period when that was just not happening. But David was looking at fundamental mechanisms of pharmacology, and he was able to keep his lab funded. And he's still around. He's retired, but he's still part of the Hefter Research Institute. So another amazing pioneer, Dave Nichols. Okay, next group of compounds I want to talk about. So the mescaline and the phenethylamines are basically confined to the cactus family. The tryptamines are all over the place. They're very widespread in plants, animals, and fungi. And when it comes to the natural tryptamines, there are only about 4 major ones: DMT, 5-methoxy-DMT, 4-hydroxy-DMT, and 5-hydroxy-DMT, which is bufotenine. These 2 are isomers of each other. The position of the hydroxy group in the 4 position on psilocin is very important, because that's what confers oral activity on it. These 2 are not orally active. They require an MAO inhibitor to be orally activated. So tryptamines are one of the most common. This is serotonin here. And tryptamines are common in nature primarily because they arise from tryptophan, which is a universal amino acid. It's one of the 20 amino acids that goes into peptide. So every organism on the planet contains tryptophan. And DMT can arise from tryptophan in 2 steps. If I can make my animation work, there we go. Decarboxylation, the removal of the carboxyl group or the acid group from tryptophan, giving you tryptamine. And then the next step, N-methylation, sticking 2 methyl groups on that nitrogen on the tail. So there you have it, voila, the simplest psychedelic biosynthetically in nature, dimethyltryptamine. And then through various additional steps, it gives rise to this family. If you hydroxylate it at the 5 position, you get bufotenine. If you come along and methylate that hydroxyl group there, you get 5-methoxy-DMT. Both of these are potent psychedelics, although bufotenine is difficultly, it has problems crossing the blood-brain barrier. It's not totally understood how psychoactive it is. But if you hydroxylate it in another position, in the 4 position, then you get psilocin. And psilocin is definitely orally active and definitely psychedelic. This is what psilocybin is converted to in the body. And then by another route, cyclization, you can take this ring or this tail and bend it around and get a 3-ring structure, and that gives rise to the beta-carboline family, these MAO inhibitors, which also have other interesting properties, which we'll talk about in a little bit. So it's not an exaggeration to say that nature is drenched in tryptamines. There are many tryptamines all over the plant kingdom. These are the families and some of the genera that these compounds have been reported from. And the numbers in parentheses are the numbers of species. So for example, acacia, one of the most, one of the families, one of the genera, there are 1,200 species of acacia. And they are some of the most potent sources of DMT and related compounds in nature. Probably 75% of those species contain DMT or related compounds. So, there are many of these things. Another, different families is where these things tend to show up. Tryptamines, because they are so common, they show up in some interesting places. For example, in citrus, some of the common citruses, such as mandarins and citrus orange and lemon and all that, they contain N-methylated tryptamines and some of their 5-hydroxylated ones. Not in high compounds, high levels. You'd have to work pretty hard to isolate DMT or one of these things from lemons from the grocery store. But if you were patient, had the right equipment, you could do it. Of course, there are many other better sources, so you wouldn't bother with it. But it's just interesting that they're so common. These Schedule 1 illegal prohibited substances are found in your citrus that you can buy at the grocery store. So here are a few natural sources of DMT and related tryptamines: the verolas, which are sources of snuffs, Anadenanthras, the human brain, of course, the various frog species, and of course the psilocybin mushrooms. So we'll take a closer look at some of these. We'll talk about ayahuasca, which means vine of the souls. It's a psychedelic beverage known by a lot of different names: yahe, pilde, natema, and it really is the most widely used of the South American tryptamine psychedelics. It's at the center of South American shamanism and ethnomedicine. And it's even been incorporated into various syncretic religious churches in Brazil, the UDV and the Santo Daime. They have adopted or co-opted ayahuasca, some might say, and they use it as a sacrament. And there has been, they actually are chapters of the UDV in the United States. that take ayahuasca, and they do that under a religious exemption due to a Supreme Court decision in 2006 under the Religious Freedom Restoration Act. So they are allowed to use ayahuasca as a sacrament because they're clearly a real religion and this is their sacrament. So the two plants that go into ayahuasca are Banisteriopsis caapi, and it's a huge jungle liana. A liana is a woody vine. It typically grows up trees. This is a plant that I planted on the Big Island in 1976, and it's now become truly the mother vine. It's about, it weighs several tons. It's like 1,500 feet long. It is in BC what we would call a skookum plant, a very, very robust plant. So lianas can get as big as any tree, and lianas typically grow on trees and eventually knock them down, which is maybe not a good thing, but that's what they do. So this plant is still there, and I planted it in 1976 on the Big Island, and then chacruna, the other admixture plant, the source of the DMT, is also growing on the Big Island. So the ayahuasca is a combination preparation made from the bark and usually the stems as well of Banisteriopsis that supplies the beta carbolines, and then the chacruna that supplies the DMT. And there are other important species too. Another important source of the DMT is Diploteris cabarrhena, happens to be in the same family as ayahuasca itself, Banisteriopsis, but instead of having beta carbolines, it contains DMT at very high levels in the leaves. So the ayahuasca beta carbolines are these three primarily: harmine, harmaline, and tetrahydroharmine. The chemists among you will probably notice that these are all the same except for the degree of oxidation of the third ring. This is the totally reduced derivative, and this is the totally aromatic derivative. And these are basically very potent and selective inhibitors of monoamine oxidase A, which is one of at least two forms of monoamine oxidase. The beta carbolines are very selective for that. And because of that, they can render the DMT orally active. Now, tetrahydroharmine is a kind of an interesting critter of the three, because not only is it a MAO inhibitor, it's a fairly weak MAO inhibitor, but it's a fairly potent serotonin uptake inhibitor. So some of the antidepressant effects and long-term effects of ayahuasca may be due primarily to tetrahydroharmine. So this just shows how this works. It's pretty simple. So you've got normally monoamine oxidase will convert DMT into indole acetic acid. But if a beta carboline is in the mix, then that reaction is blocked. So then it blocks its degradation, allows the DMT to cross the blood-brain barrier and become orally active. And of course, that totally changes the pharmacokinetics. DMT taken by itself lasts only about 20 minutes. If it's injected or smoked, it's very, very short acting. But DMT in ayahuasca will last 6 or 7 hours. So it totally, very profoundly extends the half-life of the experience, which is probably a good thing because you get more out of it. It's not so overwhelming. And, over 6 or 7 hours, you have more time to kind of get into the space that it puts you in and learn from it. So ayahuasca, particularly in Peru, is at the center of ethnomedicine. There's a bunch of people, most of them deceased now, but what I like to call the old school ayahuasqueros around Iquitos. And one of the well-respected ones that had been around for years was Don Jose Corral, This is Don Jose at the age of 99. That's the last time I saw him in 2006. So these people, some women were ayahuasqueros. Traditionally, most men were, but there are also women practitioners. It's not reserved to men. And these people, provide an important role in their communities for people that want to come to them. Effectively, they're doctors, and they know not only ayahuasca, but usually many other plants. Now, in the UDV, this Brazilian religion, the União de Vegetal, as it's called, that they call the brew that they use either Wasca, which is a Portuguese transliteration, or they call it Vegetal. And in 1993, we were able to carry out a biomedical study with the UDV at their temple in Manaus, Brazil. We got some funding to carry out this study because it had never been done before. And the UDV was, and that was mainly through the Hefter Institute. And the UDV was interested in having a study done because they wanted to demonstrate to the authorities who are thinking of banning ayahuasca, banning their religious practice, that it was not really a threat to public health, and it was actually beneficial. So we put together this group of researchers, and we went down there and did a variety of studies. We did psychological profiling, pharmacokinetics, different types of, in-depth psychiatric interviews and that sort of thing. And then we published about 8 peer-reviewed papers out of this. This study was done in 1993, and there was about 8 papers that emerged out of it. And it was very important. It persuaded CONFIN, which is kind of the Brazilian agency that's kind of a combination of the FDA and the DEA. It persuaded CONFIN to permit the use of ayahuasca as a sacrament. And then when the Supreme Court took this under advisement, this information was was important for that decision as well. So we did this study. This is people came. We did it in the temple. We didn't have a hospital, so the people would just come to the temple in the morning and drink the ayahuasca, and then we'd sit there with indwelling catheters so we could take blood samples. And some of the investigators, Dr. Jace Calloway, Charlie Grobe, Dr. Charles Grobe, and myself. At this point, I was kind of demoted to the role, I think, of chief test tube labeler, but I helped organize the study. And it was really a remarkable study, and we had some unusual findings from it. And that was really the first study on the therapeutics of ayahuasca. Before that, if you went to PubMed, which a lot of you are familiar with PubMed, the National Library of Medicine, and did a search on the keyword ayahuasca, there was maybe less than a dozen publications. But following this study in 1993, investigations began to explode. And if you go to PubMed and search on ayahuasca now, you'll find about, last time I checked, there was about 300 studies, 300 peer-reviewed papers on ayahuasca in PubMed. So another class here are the myristicaceous snuffs and pastes. The myristicaceae is the nutmeg family, and but the chemistry, these virolas, this genus, they look like nutmegs, but their chemistry is completely different. The sap of the virolas contain high levels of DMT and 5-methoxy, DMT, wait a minute, sorry, and other compounds. And these compounds, as I've said repeatedly, are not orally active unless potentiated by an MAO inhibitor. So the Indians, the indigenous people that use them, they prepare snuffs from this. They extract the sap, they powder it, and then they blow at each other up each other's nose through these long tubes, the reaction is quite violent, but it does do the job. But it turns out it's a little more complex picture, because some groups, the Witoto, for example, do prepare Varroa resin in orally active form. They prepare a paste of the partially dehydrated sap, and then they take that orally. And it may or may not include other plants that inhibit MAO. Most likely it does. But this is a rare practice, and it's a practice that's largely being forgotten. So here's a picture of the Yanomami Indians ingesting Varroa snuff from the in that form, and then this Bora shaman is preparing this orally active form, which they call ukuhe. And this is what I did half of my PhD thesis on back in the 80s, was looking into ukuhe and its chemistry, pharmacology, and so on. So another source of DMT, still on DMT, but another source of another plant source of snuffs are the Anadenanthera in the pea family. And they go by various names. And there are a couple species, Anadenanthera peregrina and Anadenanthera colubrina, and they produce these flat, shiny seeds, which are quite high in DMT, 5-methoxy DMT, and bufotenine, and they have been used as snuffs for millennia, actually. But there's a interesting paper that was published in 1981 by Manuel Torres and his colleagues, and they had been excavating shamans' graves in the Atacama Desert, and the shamans in this region were using the Sibyl snuff, as they called it. That was their name for it. And they were using them because it never rains in the Atacama, very rarely rains. All the mummies and the snuffing implements and even the snuffs themselves were perfectly preserved. So they excavated many graves, and they got the snuff tools, the snuff trays and the tubes, and actually bags of snuff, which could have been snuffed, even though they were 800 years old or more. But they didn't do that. Well, I don't know, maybe they did do that, but they carried a chemical analysis of them, and they could detect these DMT, bufotenine, and they could actually use the chemical techniques to fingerprint these samples. So they could fingerprint them according to the species, because the Anadenanthera peregrina and Anadenanthera colubrina differ slightly in terms of the profile of their active constituents. So it was a good example, one of the earlier examples of what you might call ethnoarchaeology, psychopharmacology. Many suffixes there. So and that paper was published in Current Anthropology. And then another leguminous plant in the same family is the Vinho de Urema. This is from northeastern Brazil, and it's a mimosa species, Mimosa hostilis. And Urema Vinho de Urema is an orally active preparation, so there must be an MAO inhibitor in there somewhere. And it's taken as a beverage. And it was never clear exactly what the admixture plant is, or whether there is an admixture plant, or even whether it's an MAO inhibitor, but it certainly is orally active. And recently, not too recently, but a few years ago, this interesting compound, urimamine, was isolated from Mimosa hostilis. And there's about as much urimamine in the plant as there is DMT. It's there at approximately equal levels. And this may be the secret of urema and its oral activity. The urimamine itself may be an MAO inhibitor, or maybe it's a hallucinogen in its own right. It could be a psychedelic. It's interesting that if you look at the structure of this, you can see that DMT is part of the structure. So it may be that orally, enzymes come along and cleave off the rest of it and facilitate the absorption of DMT. Just a speculation, it hasn't been proven, but that's a possible mechanism of action. So vinhodiuremia is another one of these traditional psychedelics that they're still not everything is known. You know, there's still a lot to be learned from all of these. Another group, as I mentioned, are the acacias, and these are mostly in Australia, and they are sources of some of the most potent sources of DMT and other tryptamines known in nature. Some of these are 2% DMT in the leaves. And in Australia, they're called, as a group, they're called wattles. And oddly, there's no evidence that they were ever used indigenously in Australia. Although if you look at Australian art, it certainly seems to be visionary and very reminiscent of DMT. But if Aboriginal people are using it there, they're not copying to it. They're not admitting that they did. It's interesting that wattle is the national tree of Australia. Now all the wattles are illegal. You know, the Australians are very draconian when it comes to prohibiting plants, so they actually prohibited their national tree. It can't be used because it contains DMT, and Australia is home to about 1,000 species of acacia. This particularly concentrated in this area in Western Australia. And as I mentioned before, probably 75% of the known acacia species contain DMT and related compounds. So, all right, moving right along here, we can talk about Mesoamerican toad shamanism. We've all heard of the toad, the toad Buffa alvarius is becoming quite popular because the venom contains high concentrations of 5-methoxy-DMT. You can collect the venom, you can stress the toad and collect the venom from it, put it on a glass slide, dry it out, and smoke it. The venom is about 10 to 15% 5-methoxy-DMT. And so that produces, like the compound itself, a very, very short acting, but very kind of profound and overwhelming tryptamine experience. Another species that may be implicated in this is Bufo marinus, the Colorado River toad, but it contains bufotenine, so it probably wasn't used. There are indications that toads were important among the Olmec. This is an Olmec, one of the earlier pre-Aztec, pre-Mayan civilizations. This is a ceramic toad effigy. There's no indication that the toad that is now so popular as the so-called toad medicine actually has a long tradition of indigenous use. So people that use it and run ceremonies and retreats and so on would like you to think so, but the evidence is fairly thin that it actually has any tradition of use. If it had a tradition, it's one that's been invented in the 20th century, which is okay, I suppose. And these traditions are not frozen in time, and experimental ethnopharmacologists have come away with this, and the toad medicine is it's an interesting psychedelic. It also raises all sorts of questions about the over-harvesting of the toads. They're now effectively an endangered species in the Sonoran Desert. You don't have to kill the toad to harvest the venom, but often it stresses them, so it is hard on the toad populations. But this is another area, where a graduate student who wants to pursue this area could spend their time well looking into the origins of these toad traditions. And then, of course, we come to the psilocybe mushrooms, the psilocybin-containing mushrooms. There's about 200 species of mushrooms known to contain psilocybin. Psilocybin is the 4-phosphorylase 4-phosphate ester of DMT. So it is of psilocybin of DMT. So psilocybin is converted to psilocin by removal of the phosphate, and psilocin is the active form of psilocybin. So that's rather clever. Psilocybin is rather stable, and it's what pharmacologists call a prodrug. It's the precursor of the active form. Psilocin is very unstable, will not last very, very long, but long enough to do the job. And of course, the whole story about the rediscovery of the mycolatry cults in Mesoamerica is all about traceable back to this one banker, R. Gordon Wasson, and his wife Valentina, who was Russian, who got him interested in the whole mushroom, cultural role of mushrooms. And the church authorities back in the 15th century tried to stamp out the mycolatry cults that they discovered in Central America when the conquistadors arrived. I guess it was in the 16th century, around the 1520s, and but they didn't succeed. And the mushroom cults went underground for 400 years, and then they were rediscovered by this New York banker, R. Gordon Wasson, and his wife, basically amateur mycologists interested in the cultural role of mushrooms. And then they published their findings in this impeccable peer-reviewed journal, Life magazine, in 1957. And this brought the whole thing into mass consciousness, and I think probably helped to trigger this widespread interest in psychedelics that sprang up in the 60s. And Wasson took samples of the mushrooms to Albert Hofmann, the chemist who invented or discovered LSD, and Hofmann succeeded in isolating psilocybin in 1959. And so here's a picture of Life magazine in 1957, May 13th. I had to invent time travel so that I could go back and get myself in this picture or something, maybe Photoshop, I don't know. Anyway, so psilocybin is emerging as kind of the favorite psychedelic for therapeutic use. A lot of the startup companies, a lot of the excitement about the reintegration or rediscovery of psychedelics in legitimate medicine, whatever that is, or mainstream medicine, has to do with psilocybin. And that may be because it is really the perfect clinical psychedelic. It has low toxicity, it has a relatively short duration, but long enough to be useful, to be a place where there's more time to experience and integrate it, unlike the short-acting tryptamines, which are kind of like being hit in the face and then they go away. So anyway, there are people trying to work on extending that, but psilocybin is perfectly tailored for human metabolism. It's highly visionary. It's a deep psychedelic. And because it is so non-toxic, it can be given to people in fragile health. So, many companies now are trying to improve on psilocybin, because they can patent analogs and this sort of thing. But in my humble opinion, psilocybin is just about perfect the way it is. And the mushroom is the perfect delivery system. So another group to talk about are the Lysergamides. Of course, the prototype of probably all psychedelics is LSD. LSD is not natural, it's semi-synthetic, but many analogs of LSD are found in ergot and in morning glory seeds. So LSD, of course, is one of the most potent psychoactive substances known. About 50 micrograms as an effective dose, but the more psychedelic dose, the detectable dose, 50 micrograms, that's close to what people, microdose with, maybe it's a little more than that. The usual psychedelic dose is anywhere from 100 to 500 micrograms. So it's a very potent compound. And the natural analog, ergonovine, not nearly as potent, but very similar in effect to LSD. And it has an important role in medicine. In obstetrics, it's used as an oxytocin because it can induce labor, as a lot of these indoles can do. And at higher doses, then it becomes an LSD-like psychedelic. Of course, Albert Hofmann is credited with the discovery of LSD. He was looking at a series of ergot derivatives for migraines when he happened to spill a tiny drop on his skin and experience a strange feeling of disorientation and depersonalization. That so goes the story, but it's not clear whether this is a modern myth or whether that's actually the way it happened. I don't know that we ever will, but he writes in his lab notes from April 16th, 1943, I suddenly became strangely inebriated. The external world became changed as in a dream. Objects appeared to gain in relief. They assumed unusual dimensions, and colors became more glowing. Even self-perception and the sense of time were changed. When the eyes were closed, colored pictures flashed past in a quickly changing kaleidoscope. After a few hours, the not unpleasant inebriation, which had been experienced whilst I was fully conscious, disappeared. What had caused this condition? And it has come to be known as, the day he did this has come to be known as Bicycle Day, because when he took it at the lab, he started feeling funny, climbed on his bicycle and started pedaling home as the effects were developing. And then he went back the next day. It was actually a very light dose. When he went back the next day, wondering what could have caused it. Was it maybe this LSD, this compound that I was working on? And so he went back the next day and he deliberately took what he thought could not possibly be an active dose. He took 0.25 milligrams or 250 micrograms, which, as we know, was actually a very robust dose of LSD. And then he got the full effect. Very interesting, beautiful graphic novel about this has been put out by a gentleman named Brian Blomerth. This is well worth reading. It's very psychedelic illustrations and very nice. He asked me kindly to write the foreword, which I did. So check out Brian Blomerth's Bicycle Day on Amazon and order it if you like. It's a pretty cool novel. So LSD is another fungal product. It's a derivative of these ergot alkaloids. Ergot is a parasitic fungus that grows on rye and barley and other grains, and it parasitizes it and forms these hard rusting structures called ergots, or they look like ergot means cock's spur. They look like the spur of a cock's foot. This just shows the life cycle of ergot, which, the sclerotia falls to the ground and germinates and forms these mushroom-like structures. Although they're very tiny, they're microscopic, and then they release the spores, and then it completes its life cycle. Well, in the higher plants, the Lysergamides are only found, so far as we know, in the morning glory family, the Convolvulaceae. And there are the Aztecs knew morning glories, and they called it Ololiuki. And it was used in rituals and for divination by the Aztecs. The primary species were Ipomaea violacea and Rivea corymbosa. And Ipomaea violacea is a popular ornamental. You can purchase the seeds in any garden store. When this first came out and was being discussed in magazines, the seed companies very considerately started coating the seeds with some kind of poison that would make you sick. I don't know if they still do that, but the interesting thing about these lysergamides in the morning glories is they're not from the morning glory. They're actually from a fungal endophyte that lives inside the morning glories. And this is very common, that there are fungi in plants that produce complex secondary chemistry. It's not always because of the plants. And in the case of the psychoactive morning glories, it's because of these fungal endophytes that produce these ergoline alkaloids. And probably the most potent source of these compounds in the morning glory family is the Hawaiian baby wood rose. The other ones, you have to eat about 600 of these tiny seeds to get anywhere. You'll get sick long before you've eaten enough. These you can get away with 8 to 10 seeds or maybe 15 seeds. And so these Hawaiian baby wood rose, which are neither Hawaiian nor a wood rose, are a fairly good source of the psychoactive morning glories. And but it's only one of about 90 species. And these Argyria species do have medicinal uses in Ayurvedic medicine, but no evidence that they were used for psychoactive effects, although certainly it had to have been noticed. Somebody was probably doing it, but it's not really in the literature. So let's see, where are we at with our time? We have about 25 minutes left. Maybe I'll stop this and see. So people, do you want to go on or do you want to stop here? I could go on and discuss some of the non-psychedelic but shamanic plants. That would probably take up the rest of the half hour, or we could stop here and discuss, or what would you like to do? Who is running the show here?
Yeah, why don't we break for a few minutes? We can answer questions we've got right now, and then if that settles, let's move on and do more material.
Okay, what time does the class officially end?
6 o'clock is when we schedule the end of our lecture Q&A. Technically, we have the space for a bit longer, so it's not a big deal if we go over that.
All right, well, is that a good idea? Should we do a bit of Q&A and see how it goes, and then we can start again?
Yeah, sweet.
Okay, all right. I'm going to put a link in here, which we may not get to, but this is the videos from the conference I did in 2017, the Ethnopharmacologic Search for Psychoactive Drugs 50th anniversary. People can go and look at these videos if they want to review those presentations. They're still up there. I may get to talking about, we have another one coming up in May. I may get a chance to tell you about it, or maybe not. But anyway, there's that link just for you to keep in your whatever, on your bookmarks. So we can do some questions now, and then we'll see how the time goes. I thought, yeah, I tend to be long-winded. I thought we'd have more time. But so is there anyone with any burning questions? I guess you can raise your hand in the reactions, or just put it in the chat box, whatever you'd like to do.
Yeah, and if anyone in person has a question, just raise your hand and we'll call on you, come over. Yeah, Andy. Andy I and then Andy Y.
So should I just like,
Yep.
Okay, lovely. Hello. First of all, thank you very much for your presentation. This has been very interesting so far. So it's kind of a niche question. You mentioned the kind of mushroom cults, so to speak, in Mesoamerican cultures regarding magic mushrooms. Why do you use the terminology of cult? Is there a specific reason for that?
Of cult, well, yeah, maybe it's a loose use of the term. I mean, they are religions, you know. They're maybe sect is a better word. It doesn't have the pejorative notion, but these were basically mycolotrous cult, you know, mycolotrous religions. They worship the mushroom, and you know, so. But yeah, maybe the term cult is not a precise use of that term. I mean, is the Catholic Church a cult? Some might say so, you know, but we tend to think of cults as small and insular. And good question, you know, it doesn't really apply because the use of these mushrooms in Mesoamerica back in, you know, before the conquest, before the conquistadors got here, was actually quite widespread. You know, the Mayans, the Aztecs, the Mazatec, various groups used these mushrooms. So it wasn't a single cult, it was various religions that use these things.
Awesome, thank you.
Yeah, thank you.
Then, yeah, I think Andy and then anybody else.
Hello, Dr. McKenna. I had a question about the myristiaceous snuffs and the other DMT-containing snuffs. Is the experience and duration similar and comparable to, I guess, vaporizing compound?
Is it similar to DMT?
Mhm.
Well, I can't necessarily tell you from personal experience. The experience is similar to DMT in that it is very short, you know, and it's not as DMT is normally smoked, right? You don't smoke the snuff, you snuff the stuff. And the way the Yanomami do it, they do it through a long tube, you know, a 3-foot long tube. One person blows it into the nose of the other, a couple of grams, and you know, and I think there could be nothing in the snuff, and you'd still have a violent reaction, you know. And they do violently react. I mean, the people that receive it, they kind of, you know, fall, you know, they start emitting snot all over, they fall down, they twitch, they're going through a kind of seizure, and then they'll get up and start to dance, you know. And that's when they, you know, these people are tough, these people are bare wires. So, you know, smoking or vaping synthetic DMT is a lot easier way to do it, you know. And yeah, so they're definitely active, but they're you know, they're hard on the nostrils, that's for sure.
Thank you.
Yeah.
Yeah, go ahead. Yeah, maybe ask and then sit down. So you have both of you.
Hi, thank you so much for your talk. I was just wondering, so you mentioned how ayahuasca's like beta carbolines act as MAO inhibitors, and so I was wondering, to your knowledge, like, does the amount of MAO inhibitors that you need to overcome the MAO like enzymes depend on how much DMT you take, or do you just need to have like enough MAO inhibitors to like just block all of the enzymes, and then like how much DMT you
Well,
have?
you need the MAO to inhibit the breakdown of the DMT. And so depending on how much DMT there is in the brew, you could still get, you know, a very strong experience or a very light experience, depending on, you know, how much of a dose you take. In taking ayahuasca, sometimes people will take just a little bit. It's very common taking ayahuasca that you would take a booster dose. You could kind of titrate the dose that way. But the MAO has to be there for the DMT to have oral activity. And then how potent it is, I guess you could say, is going to depend on how much DMT there is. And these are not exactly pharmaceutical, you know, standardized preparations. There are many variations. Depend almost as many variations as there are practitioners. And there are also many other admixture plants that are sometimes used. We didn't really get into that, but the basic formulation is, the Banisteriopsis vine, and then the leaf. that contains the DMT. Other plants may be added sometimes for special purposes. So that's it. It's a detailed thing and not completely worked out. There are about 150 species of plants that are sometimes used in addition as additives to ayahuasca, along with the two main ones. So that changes the effects, of course. So this is another area that really needs to be explored by experimental ethnopharmacologists or ethnopharmacologists. There's not everything is known about ayahuasca. There are a lot of questions that still need to be answered with that one.
Thank you.
Okay. So I could talk about the next section, which is somewhat shorter, mercifully, or what would you like? What would you like to do? Someone else wants to ask a question there.
Yeah, we've got just about 2 burning questions, and then we can get through those and then hop back to the lecture.
Okay, sounds good.
Awesome, great.
Yeah, hey Dennis, I got kind of a maybe unrelated question. So let's say I have 12 monkeys and 5 grams of synthetic psilocybin. How do I go about testing the stone ape hypothesis?
Yes.
Neither of those premises are necessarily untrue, by the way.
So how do you go about testing the psilocybin?
The stone ape hypothesis. If I have 12 monkeys and 5 grams of psilocybin, how would you test it?
I'm not sure what you mean. I'm not sure I'm understanding you completely.
Sorry.
The stoned ape hypothesis, if you had 12 monkeys and a bunch of mushrooms, how would you go about seeing if you could validate or disprove?
Whether they work in the 12 monkeys?
No, whether the stoned ape hypothesis, whether it's true.
I don't know. I can't answer that question. Sorry.
Okay. Yeah, I assume you're familiar with the stoned ape hypothesis.
Okay, yeah, it's way beyond my pay grade. I don't know. I'll have to pass on that one. Sorry. Another question?
Yeah, we had just one more. Yeah, right? Cool. Yeah.
Okay.
Hi. So why do you think plants produce the psychedelic inducing effects? Like, what is beneficial for the plants about sending these messages to humans and animals?
Well, that's a complex question. Like I say, the plants use these compounds to mediate their relationships with other organisms in the environment. So they can use it to initiate symbiosis, for example, and they can use it to initiate symbiosis with humans. And for the plants, psilocybin, for example, probably didn't evolve to make humans high, or to delight humans. Psilocybin in mushrooms probably existed. In fact, we know that it existed. Mushrooms have been around for about 75 million years. That's when they branched off from other branches. And the basidiomycetes, the advanced mushrooms that contain psilocybin, probably had these compounds many millions of years before there were primates or any kind of complex nervous system for them to interact with. And there's now plenty of evidence that actually they were evolved to mediate insect interactions, to either be repellents for insects, more likely to attract insects, to assist in spore dispersal. Fungi and plants, they want to grow, they want to spread. This is their mission. So there's interesting studies with psilocybin, and for example, in cicadas. I can send you some articles about that. Basically, mushrooms evolved psilocybin to drive insects crazy, But then when we came along, those compounds became adapted to a different kind of symbiosis. We found that we hominids way back when found that we enjoyed these experiences, and so the symbiosis was formed that way. Essentially, we began to use the mushrooms, and because the mushrooms were associated with cattle, right? They grow out of cattle dung, so there's something that was just around the village, or they didn't even have village, nomadic peoples, but probably in that process, once the properties of the mushroom were discovered, we formed this coevolutionary symbiosis. You have to Google the stone date theory, and it will come up. I will send a little clip to the instructor, and I can share it with you. You can find it. There's an interesting piece on YouTube about this. But this is what plants use these compounds for, not necessarily the psychoactive ones. Any plant that we interact with, whether as food or medicine or for psychoactive purposes or whatever, it's usually traceable to some chemical property. Maybe it has to do with taste or nutrition. All of these things can be bases for these symbiotic relationships, which are chemically mediated. So that's what these compounds can do, because again, if a plant can ally itself with primates, with humans, effectively it's affected a sort of evolutionary free ride. We've taken it doesn't have to compete out in the wild if we domesticate it. Of course, there may be a downside to that for the plants or the mushrooms in some respect, but usually that's a beneficial thing. So symbiosis, there are different kinds of symbiosis, but the kind we're talking about is commensalism or mutualism, where both partners benefit from the relationship. And the mushrooms benefit because we grow them, we spread them around. We benefit because they provide this compound that opens the vision screens to many worlds, which we seem to like. Does that make sense?
Great answer.
Sometimes it makes sense. Okay. Shall we continue? I'll see how far we'll get with this.
Sure. I want to try to clarify one of the questions we had. It was about the stoned ape hypothesis, and just the idea of how could somebody try to investigate that with like a reason.
Yeah.
Okay.
Because I have monkeys and a DA license, so I would be in the position maybe to do it.
It's difficult to approach that experimentally. I don't know that you could learn much from doing that. What you have to do is, I don't know how you'd explore how you would approach it experimentally, because we're talking about a relationship that happened so far back, that there's not going to be any evidence, except that we know that the climatic conditions in Northern Africa, where hominids evolved, Northern and Southern Africa, were much wetter, much more tropical. They were savanna environments, but they were wet. We know that there were cattle there, or cattle-like animals, because we have the fossil evidence for that. We know that there were hominids there because we have the fossil evidence for that. So very likely, as these hominids evolved in this environment, the mushrooms, and we're talking about Psilocybin cubensis, which is the pan-tropical, large, robust psilocybin mushroom that grows out of cattle pies, cow pies. So if you're a hominid and you're foraging in this environment, you would have to be blind not to overlook this thing. And if it's there, even if you don't know about its properties, you're going to eat it, right? Once you eat it, that connection is made. And then, so that's probably how the discovery was made. I actually have a whole lecture on this topic, which I could give to you at another time. But yeah, I think it's, I mean, people will say that's a crazy idea, but it's actually more than likely, and there's a couple things about it that make it more plausible that we have learned since my brother originally sort of proposed this idea. One of those is neuroplasticity. We know that the psilocybin can increase the connectivity of the neural architecture. It actually complexifies the neural architecture. And this may contribute to the rapid evolution of the human brain over a relatively short period of evolutionary time, about 2 million years. And the other mechanism here is epigenetics. Epigenetics is a way that these environmental influences can persist through generations without invoking the usual mechanisms of inheritance. It doesn't have to do with mutations. It has to do with the internalization of through maternal inheritance and other mechanisms of these changes, which can actually become heritable across generations. But I can't get off on this tangent too far, or we'll never get out of here. But I'd be happy to come back and present that presentation to you at another class. But that's it, kind of in a nutshell.
Right on.
Okay.
Just one comment. Like I think of it as less often like evolutionary mechanism, because it's kind of hard to inherit it through, as you say, through mutation. Well, epigenetics are not clear either, but I could imagine that maybe within the lifetime of a monkey, he could have some kind of insight from it, and that through culture is then inherited. If he figured out how to suddenly develop language or some primitive form of it.
Yeah, but the thing is, these are things that happened over many, many generations, I think. I don't think you could, I don't think you could feed mushrooms to a group of 12 monkeys and suddenly they invent language and culture. I mean, I don't think it happens that quick. I think it takes, generations for these things to, to manifest. So, but yeah, we're way off track here right now. So I'll share with you a couple of clips that I think will shed some light on this for you.
Thank you.
Okay.
Yes, we have just a few minutes. If there's more lecture material that you'd like to move through, or anything else that you want to share with the class, space is yours.
How long do we have?
We've got 3-4 minutes until 6, and and plus a few more minutes if we need.
Okay, well, I tell you, I was going to go through the second section here. Wait a minute, I want to share that. And then I want to, okay, so the second section was called shamanic, but not psychedelic. And I don't think there's time really to go through that. So what I am going to do, actually, because I want to let you know about this conference that I am planning that is in progress, and we'll have to wait for the shamanic but not psychedelic section. And then I had another section called bitter brews and other abominations, just looking into some very sort of esoteric corners of nature for potential psychoactive drugs. But what I wanted to share with you that may be of interest is this conference, which I organized in 2017. It was a 50-year follow-up of a conference that was originally given in 1967 sponsored by the National Institute of Mental Health. And all they ever did was publish a symposium volume out of this. They were supposed to have follow-up conferences every 10 years or so, but it never happened. So 50 years went by, and the book that came out of it was very influential to me. So I wanted to organize a 50-year commemoration of the ethnopharmacologic search for psychoactive drugs. So I did. Everything came together in 2017. It wasn't just me. I had lots of help. I don't want you to think that I did all this by myself. But it was a very successful conference, and we published the symposium proceedings as a box set, and we also published the original 67 volume in the set. So, we published this, and that clip that I pasted into the chat box at the start of the lecture is all the lectures from this 2017 symposium, right? So you can go there and listen to those lectures, many of which are very interesting. Well, now we're doing a follow-up. We're doing a 5-year follow-up on that. So we're doing a 55th anniversary symposium, and it's going to be a 5-year follow-up in the tradition of the first one. I mean, when the government did the first conference, they were supposed to follow up every 10 years or so. But the war on drugs came along, and they got embarrassed that they had anything to do with. So we've taken up the gauntlet, and you can go to ESPD 55 and see about this conference coming up in May, this May, except that the website isn't quite open yet. We're still working on it, but within the next 48 hours, it should be open if you want to see the program and speakers for this event, which will be live streamed. It'll be in the UK, but last time we live streamed the conference, we had 75,000 people watching in live stream. Hopefully we'll have a comparable number for ESPD 55. So I guess the message here is stay tuned if you're interested, and you can watch those videos from ESPD, ESPD 50.